Welcome to Birch. My name is Brian Frstensky. For those starting out in bioinformatics, one of the first things you need to learn about is how DNA and protein sequences are represented in data files that are read by the programs you use. There are a seemingly endless number of formats, but today I'll cover just a few of the basic formats needed for working with sequences. And these will include the most common ones like Fasta and GenBank and Swissprot, along with a few others that you've probably never heard of before. You won't be surprised to learn that nucleotides are represented by their one-letter codes, but you may not know that there's an exhaustive list of codes covering all the possible nucleotide ambiguities. So we've got the familiar G, A, C, T, and U for the four nucleotides, and many of you have seen R for purine and Y for pyrimidine, but there are other codes for the different possible ambiguities. So K for G or T, W for A or T, V for G, C, or A, and then finally N for any of the four nucleotides. Now, for amino acids, this is a little more complicated. There's both a three-letter code and a one-letter code. Human beings are much more familiar with the three-letter code. It's easier to get which amino acid you're talking about. The one-letter code, though, is necessary for computer programs because it's way easier to write a computer program that can read single letters for each amino acid. So F is phenylalanine, M is methionine, P is proline, Y is tyrosine, and the stop codon is represented by an asterisk. X stands for the unknown amino acid, so it's a placeholder if you need it. There are also codes for the ambiguity of leucine or isoleucine, which is represented by the letter J. And some prokaryotes actually have an unusual amino acid called pyrrolysine. And if that's included in your genetic code, then O would stand for pyrrolysine. So the first format we'll start with is just the raw sequence file with nothing but nucleotides or amino acids in it. So I'm going to show you an example now where we have an actual file and we can show how a program would read such a file. So here's an example of a raw sequence file, and you may note, by the way, that I'm looking at this file in a program called Pluma, which is an example of a text editor. So some of you may or may not know that a text editor is a lot like a word processor, but it only works with plain text, letters, numbers, any character that you can see on the screen. There's no such thing as fonts or boldface or underlining or pagination. It's just plain raw text. So most data for scientific work actually is in text files of this type, and so you read these files with a text editor. On Windows, you might think about, for example, the Notepad editor as an example of a text editor. So if you've never written a program to read a file, you might not know that the way files are read is starting with the first character on the first line and going across the line till you get to the end of the line and reading one line at a time until you keep going down and get all the way to the end of the file. So this is what a program would do if it was reading one of these raw files. They just read in all of the characters, and then they would be read into a single long sequence. The line breaks, the breaks between the different lines are not a part of the sequence itself. By the way, these numbers on the left are inserted by the text editor. They're not actually part of the file. So the only thing you see that's actually in the file is what's shown here in green. So this is an example of a raw sequence file. Not too many programs use them, but sometimes it's useful to have a raw file like this. So very similar to the raw sequence file format is a format used in the Birch system that we call free format. And by the way, on these title lines, I'm including suggested file extensions. These are just suggestions. There are not always conventions for how you name files, but these are good ones to use. And so the free format is very similar to the raw sequence file in that anything other than a legal nucleotide or amino acid character is read, but so things like numbers and spaces would be ignored. But there's another rule, which is that you can put comments into the file so that anytime a semicolon is seen, that begins a comment and the rest of the line is ignored. Probably the most common file format that you're going to see is called the Fasta format. And so this is a little more sophisticated format in the sense that it gives us a way to include a name for a sequence and also to put more than one sequence into a single file. So I have here an example of a Fasta file that has two sequences in it. And at the top I've got a kind of a formal definition. So there's every sequence has two components, a name line and then one or more sequence lines. So the name line always begins with this right arrow character followed by the name. And I'll make a special point, by the way, the right arrow character is not part of the name. It's just there to tell a program that's reading through the file, this is where the name line starts. And sometimes there's also a definition that goes with the name, and that would be anything after the first blank space after the name. So the name is assumed not to have any blank spaces. It has to be letters or numbers, but no spaces, no tabs or anything like that. The definition is just a human readable part of the line. And then after the name line, there are one or more sequence lines. Some Fasta files will have a big long line, maybe thousands of characters long, or more than that perhaps. Others will break the sequence up into many lines, and there's even some variation in terms of some programs will only read the two-line Fasta sequence. Most probably will read this multiple format though. And so you can see here that there are two sequences, and I've even got a little algorithm here to explain how these kinds of files are read. So you keep repeating this process. Read a name, then start reading a sequence until you get to the next name, read the name, then start reading that sequence until you get to the end. So because we have a way of breaking up the sequences with these name lines, Fasta files can store more than one sequence, and in fact sometimes millions of sequences in a single file. So here's a Fasta file, the one we were just looking at actually, in a text editor. I'm just going to make this wider so that the lines don't wrap. I guess I'm going to have to make this pretty wide so that the lines don't wrap. There we go. Sometimes you'll get that in text editors. The line is long and it wraps around to what looks like the next line, but really it's all one line. So if a program was going to read this, it would see the arrow character and then it would get the name. It probably will ignore the rest of the definition line, and then it'll read the sequence until it starts encountering an arrow so that it says, aha, we've gotten to the next sequence, we're going to read the name for that one, and then we'll proceed to read that sequence. And so, as I've mentioned before, the Fasta format is a big improvement over the raw format in that it lets us have as many sequences as we want in a single file. So even a Fasta file doesn't really let us store very much information about the sequence. So for that reason, NCBI created the GenBank format. And the GenBank format has a very rich collection of annotation for any sequence, followed by, of course, the sequence itself. So these can be fairly large files. And what I want to do next is go through the structure of a GenBank file. But before I do that, I'll just mention that if you're really interested in the nitty-gritty details of the formal definition of GenBank files, I have a link at the bottom of this page that will take you directly to the current release notes for the GenBank database, and it includes quite an exhaustive and precise formal definition of the GenBank format. So as you can see, there's quite a lot to the GenBank format, and so the best way is just to start at the top and proceed downwards. So there are keywords in the left-hand column that tell you what part of the file you're looking at. And so the first line is the locus line. And the locus line begins with a name for the sequence, and most often the name is exactly the same as the accession number, but there are some exceptions. And then we have the length of the sequence, usually in base pairs. We have a field telling us the type of sequence. Now, this is a DNA sequence. Usually DNA means a genomic DNA. You could have mRNA, tRNA, rRNA, ncRNA, siRNA. There's a bazillion types of things that could be in this field. Then you have the topology. Most of the time it's linear, but if you had something like a plasmid or a mitochondrion or a chloroplast genome, then it would be circular. There's also a three-letter code for the division. So there are quite a lot of divisions in the GenBank database. This sequence comes from plants, so it's from the plant division. There's a vertebrate division, VRT, a viral division, VRL, a phage division, PHG. If you go to the documentation for GenBank, you can find what all those are. And then finally, the date on which the sequence was last updated. Then there's a definition line. This is just a quick summary of what's in the file. And then the accession number. So the leftmost accession number is always the current correct accession number, but sometimes there are secondary accession numbers which are older numbers. The main point of an accession number is that when you publish a sequence, even before you submit the paper, you're required by just about any journal these days to submit your sequence to GenBank and get an accession number for that sequence. And then that accession number is included in the manuscript when it's reviewed, and in the manuscript, of course, once it's published. But the main point is that that accession number is unique to your sequence. No other sequence will ever get that accession number, and so your sequence can always be retrieved by that accession number. Now, at some point, there must have been an update to this sequence, and so they assigned it a new accession number. You can still retrieve it from the old number, but the new accession number is the current number. There's also a version of the accession number if that's necessary. I can't think I've ever seen anything other than .1, so that does exist, but don't worry about it. Keywords, it's pretty self-explanatory. The source information gives you the full taxonomic hierarchy to which the organism belongs. So this sequence came from cotton, Gossypium hirsutum, and it's a sequence for chlorophyll A binding protein gene. And so this is in the light harvesting complex 2 in plant chloroplasts. There's usually literature citations, and then we get to the features table. So the features table is often the biggest part of any GenBank entry, and as you can see, we have the keyword features and then the location/qualifiers field. So for every important feature in the sequence, there will be some annotation telling where that feature occurs. And every feature, by the way, also includes a source feature, which includes the entire length of the sequence. If you needed to be able to refer to that entire sequence, you have a feature that does that now, and it will contain other information. So you can see here information on the library and that kind of thing. If we scroll down a bit, you'll see a gene feature, and you'll see here that this CAB gene begins somewhere upstream from 454 and ends somewhere downstream from 341. That's literally what these arrows refer to. So why do we have the arrows, the less than and greater than ones? We'll show you right now. If you take a look at the coding sequence, the CDS, so there is the coding sequence feature, and you'll see that that begins exactly at 454 with no arrow and ends exactly at 1341. So the 454 in this case is the first nucleotide of the start codon. 1341 is the last nucleotide of the stop codon. And uh, we have some additional complications. You can see it's actually broken into two parts. We have a join command to join these two parts together. And why is that? Because you have two exons separated by an intron. So the first exon begins at some 454 or somewhere upstream, and ends at 497. The intron goes from 600 to 597. The intron goes from 600 to 689, and then the exon 2 goes from 691 to 1341. mRNA is of course also annotated in the same way, and normally in a genomic clone you'd think, well, the mRNA is going to include some 5' untranslated region and also some 3' untranslated region. And that isn't the case here, simply because although those things undoubtedly exist, the work was not done to figure out where the RNA starts and where it ends. If you had a cDNA clone for the same gene, you might almost be able to get the start position, although most cDNA cloning methods actually truncate the 5' end, so you're still missing some of the 5' end. So the real take-home lesson from all of this is that most coding sequences and mRNAs are actually not the complete sequence. Or actually, I should take that back. Most mRNAs are missing some annotation or some designation of where the RNA starts and stops. The coding sequence is undoubtedly correct, but the extent of the entire RNA is not. So then, I give this as an example because it's important to see these kinds of somewhat awkward things, because you're going to encounter them all the time in the databases. There's another format that's very similar to GenBank, which is the EMBL format, and this has all the same information, but it's a little less wordy. You can see, for example, the keywords on the left are all exactly 2 characters, and then the rest of the information is out by itself in a little more friendly form to the human eye. And so probably fewer programs read this format than GenBank, but it is something that you may encounter. By the way, I should mention that these more elaborate file formats that I'm talking about, GenBank, EMBL, Uniprot, SwissProt, are really not how the data is stored in the databases. The databases will store the information in a very efficient relational database system, and the flat files that you get in GenBank format, EMBL format, whatever format, are best thought of as reports generated from the database. So if you retrieve a GenBank or an EMBL file from one of the online services, that file didn't exist until it was created in response to your request. So at that time, then the file will be written in whatever format is specified and then sent back. So that's just interesting to know about these formats. I should also say that the more elaborate formats of this type are not read by all sequence programs. Most programs probably work with Fasta files, and so that's probably the best default assumption, but you have to check the documentation for the programs that you use to know what kind of format they need. So I'm sure you won't be surprised to learn that there's an equivalent format for protein sequences, and this is the Uniprot or SwissProt database format. So SwissProt is the old name of the database. It's more recently known as Uniprot, and it has highly curated protein sequences from many different sources. The gene that we're looking at here, by the way, is the same or I should say the protein. This is the protein encoded by the chlorophyll A binding protein gene that we were looking at before. So there's a one-to-one correspondence between this protein and the GenBank entry that reports the entire gene. So I want to take a look at a typical Uniprot entry just to give you a sense of how that looks. So here's the Uniprot or Swissprot entry that we were looking at just a moment ago, and again it has a locus line and the number of amino acids. I don't know if there are any circular proteins, but this one certainly is linear. And we have a definition line, much the same. The accession number. So the accession number is a protein accession number, which is distinct from the DNA accession number. So this is the Swissprot or Uniprot accession number. We'll just scroll down. Most of these things are the same. Sometimes there are very elaborate comments that tell you a little bit about the biology of the protein, and that's the case here. And then we have a features table. Once again, there's a source feature for the entire sequence, and I'm surprised actually if there's a gene feature in a protein entry, but whatever. And then the protein, you have different regions. For example, whatever, this is a mature chain, so after processing of the protein, sites for things like acetylation or phosphorylation or metal binding sites, all the things that you might expect to see for proteins. And then at the end, it's almost like an afterthought, isn't it? I always look at these and say, boy, the protein sequence is so small compared to all the annotation that there is about the protein. And so this is, again, a fairly elaborate file format for proteins. Most programs that work with amino acid sequences expect you to give them a Fasta file and not a Swissprot file. Having said that, I just want to point out that part of the rationale behind the BioLegato programs in Birch is to save you the trouble of having to interconvert file formats all the time. So if you read in, for example, a GenBank or Swissprot entry into one of the BioLegato programs, BLDNA or BLProtein, it will know the file format that's needed for any program that it runs, and it will convert the sequence into the appropriate file format when it runs the program. So this saves you a tremendous amount of time not having to manually create files in the formats that you need, let alone having to go look up the documentation and see what kind of file format does this need. You have better things to do with your time, and so BioLegato actually saves you a lot of effort and a lot of error. So what I've told you about today is really just the tip of the iceberg. There are more sequence file formats than you can count. Next generation sequencing programs will write millions of reads into files called FASTQ files that have both the raw sequence and quality information. Genomes tend to be written in such a way that the DNA is split into one set of files and the annotation into another set of files called GFF files. There are formats that map reads to genomes or give you the location of sequence variants. When you start getting into more genomics tasks, you will have to start worrying about these file formats. But for now, these are some of the basic ones that would let you work with sequences. I just want to remind our listeners that Birch is free open source software that can be downloaded at the address above. And because it's free open source software, the way you pay for it is by citing the publication that it comes from. And I hope you cite all the bioinformatics software that you do. And so please check on the Birch Bioinformatics YouTube channel for more videos on bioinformatics using Birch.